tell you a story that is about 40 years in the making. In fact, I was a graduate student at the time that I first stumbled into this project, and it was a few experiments as a graduate student that later snowballed into a career. So I guess for students in the audience that may keep your eyes open, there could be something close by that could make a big difference in what you want to do. So this is a figure from a symposium that was held at Cold Spring Harbor in 1960, and it gives a sense of where the thinking was at the time with regard to the biological clocks that govern our behavior and the behavior of so many plants and animals around us. This is work that a scientist whose name was Pat DeCourcy spoke about at the meeting, again, 1960. And nothing's changed with regard to what's shown here, which is the phenomena that makes you wonder what is behind this kind of rhythmicity. This is a rodent in a cage, with a running wheel, and this experiment is performed in complete darkness. The experiment lasts 25 days, and each day of the experiment is represented by a successive horizontal line, day 25 down here at the bottom. And this is a 24-hour, each one of these lines is a successive 24-hour interval. The thick parts of these lines are generated by the movement on the running wheel in the cage by this, it's a flying squirrel that she was studying. So what you see is that on day 1, the animal gets on the running wheel. Day 2, the animal gets on the running wheel. No environmental changes whatsoever. Constant darkness, constant temperature. And yet, within a few days, you realize that you can predict when the animal's going to get on the running wheel and off the running wheel with an accuracy of 2 or 3 minutes. The other important feature about this is that this is not a vertical record. The record runs out to the left, indicating that the periodicity of this behavior is not exactly 24 hours. It's less than 24 hours, which certainly must mean that something internal is driving this pattern. There's not some astrophysical information that's being picked up by this animal to produce this behavior. This must be internally driven behavior because it's not, it doesn't correspond to any 24-hour external physical part of the world. Now, in trying to understand what this is coming from, in the 1970s, scientists began to look within the brains of these rodents for altered metabolic activity that might indicate physiology that was changing in a night-day kind of fashion. This is work that was performed again in the late 1970s, in which William Schwartz, who's now, I think, at the University of Texas, one of Gail's and Steve's colleagues. These are serial sections of a brain from a rodent that is given radioactively labeled water. That is radioactively labeled because there's sugar in the water, glucose in the water that's radioactively labeled. And what he noticed is that this region at the base of the brain and part of the hypothalamus called the suprachiasmatic nucleus, which we can just call SCN, is lighting up in the middle of the day, but can't be visualized in the middle of the night. So there's a strong oscillation in the metabolic activity of this region of that rodent's brain. Now, when this was seen, additional experiments were done in which that region of the hypothalamus was surgically eliminated. And what was found is that the animals could no longer show the rhythmic behavior that I demonstrated in the prior slide. So they became arrhythmic without this, without the SCN. But even more remarkably, scientists realized that they could borrow the SCN from a fetal animal and replace the ablated SCN in an adult and restore rhythmicity, indicating that not only is this an essential tissue for this kind of regulation, but that that regulation could be mapped in some way to the activity of that piece of tissue. Now, this was all occurring as I was starting graduate school. And in 1971, just as I was beginning my work, this paper was published in the Proceedings of the National Academy of Science by Seymour Benzer and his student Ron Konopka. And they had isolated the first mutants, genetically defined mutations, that would affect this behavior in any organism. And these happened to be mutations that were acquired in Drosophila. The mutants were extraordinary, not only because one class of mutants would eliminate this rhythmicity, just as an SCN lesion in a mammal would produce arrhythmicity, but another mutation produced 19-hour behavior. That is a fast-running clock of some sort. And a third mutation, a long-period clock, a slow-running clock. So you had 3 different mutations that affected these rhythms in 3 different ways, and the most remarkable piece of this was they all, by genetic mapping, seemed to be alleles of the same abstract genetic locus. So this suggested to me that there could be a gene which could be altered in at least 3 different ways to speed up this mechanism, to slow it down, or to obliterate it altogether. Now, as I mentioned, they all seem to be, by genetic tests, part of the same functional locus, but there was only rough recombination mapping that suggested that they also were located at a location on the X chromosome of the fly, but it was ill-defined. Now, this was a lucky find for me because at the time, I was studying for different reasons the region of the chromosome shown here. At the top of the chromosome, at the top of the slide is shown a diagram, a drawing of one of the giant salivary gland polytene chromosomes that you find in Drosophila. These have been used for almost 100 years to localize genes and gene activity in the fly, dating all the way back to Painter, who was a professor at the University of Texas in the 1930s. And before him, Drosophila had been used for genetics for discovering that genes were on chromosomes by Thomas Hunt Morgan in the early 1900s. So there was a long history of using these Drosophila and these chromosomes to locate gene activities. Now, at the time, I was interested in a region containing about 20 of these dark bands. Each of these bands seemed to be associated with a single functional unit defined by mutation and genetic complementation, which is allele tests. And I had been trying to understand whether or not there really was a one-to-one correspondence of the functional units in this region and these striations, these densely DNA-rich regions of the polytene chromosomes. When the Konopka and Benzer paper came out, I realized that with some preliminary recombination mapping that gene seemed to be mapping somewhere in this interval. I'd gathered a number of chromosomal rearrangements, translocations, deficiencies, and duplications in this region, and thought perhaps I could use those to better locate the gene. Now, what you see up here is a designation called PER. That's period. That's the name of the gene we've been talking about. What I realized by putting all these rearrangements together was that PER fell right in this location, which could be surrounded and even broken by some of these gross chromosomal changes. And in particular, I found one translocation, which is shown here as JC43. This is a reciprocal translocation in which the tip of the X chromosome has been moved to the 4th chromosome, and the tip of the 4th chromosome has been moved to the X chromosome. No missing genetic material, but that translocation had broken this gene period and gave an arrhythmic phenotype. So this was the 4th mutation to be found at the locus. But this is a a very special mutation because in the late 1970s, early 1980s, there was no large-scale DNA sequencing. If you wanted to find a gene, you needed something that altered the map in a severe fashion. And this translocation is something that if you had cloned DNA, if you had isolated DNA, you could map a rearrangement breakpoint and locate the gene. And this is something that I realized several years later when I was at the Rockefeller University, thinking once again about this work that I had done as a graduate student that had been published several years earlier. So in 1984, my lab and the lab of Jeff Hall and Michael Rosbash, colleagues at Brandeis University, used those rearrangements to locate DNA from this region of the Drosophila chromosome, and then to use a transformation experiment to move that DNA back into animals that were deficient for the period locus and prove that indeed we could rescue the behavior of an arrhythmic mutant by taking DNA from a wild-type fly from this area and moving it into embryos and allowing those embryos to form new adults. So in a sense, this is very much like those SCN transplantation experiments. Instead of transplanting a tissue, though, you're transplanting a gene, and the hope in this case was that by following these genes, you could really understand the molecular mechanism behind this kind of behavior. So that was the hope. Now, this was the first gene to be isolated in this way, and looking at the data, we realized that this was all very exciting. It suggested there was a molecular mechanism that might be approached, but what molecular mechanism do any of us know about that is controlled by just a single gene? We know that a single gene mutation can affect the mechanism, but it doesn't describe the workings of the mechanism. So we realized we needed to do more work. We needed to go back in. We needed to isolate new mutations that would affect this behavior. And we did an experiment that looks like this. Now, this is a Photoshop recreation of the very second mutant hunt in the fly. 7,000 bottles of flies. Amita Segal and Jeff Price, two postdocs in the lab, had to look through every one of those bottles, not just once, but every 4 hours for a week, to determine whether or not there were any flies in those bottles, in any of those bottles that had an aberrant form of behavior, hatching behavior. Flies hatch from their pupal cocoons in the morning if they have a normally running biological clock. And in the 7,000th and something bottle, Amita and Jeff found a new mutant. And that mutant was arrhythmic. It looked very much like the arrhythmic period mutation that I described before. Caused arrhythmicity not only of the eclosion pattern, of the hatching pattern, but also of locomotor behavior, which we'd seen as well with period. So we had a phenotype that seemed to be identical to that of period, but now coming from a new gene that was located on a different chromosome. Now, this turned out to be extremely useful to us because as we did additional molecular studies that I'll tell you about in a moment, we realized that this second gene, which we'd just been asking, can this system, can the fly point us toward a second gene that affects this mechanism? Turned out that the gene that was identified is a physical partner for period. And so not only had we found another gene, but we had found another piece of a mechanism in which two components work together, which gave us two pieces of information. It suggested, first, that we were going to find additional components in this mechanism, but second, that the mechanism cannot be huge. If the very next hit that you have is in a gene that produces a product that works with the first component, then the size, the target of this mechanism can't be overly large. So we were very excited about that, but we didn't want to do this again. I was happy to do this again, but Jeff and Amita were not. So we resorted to a different approach. This is a mechanism in which you have this device connected to a computer. Each one of these is a glass vial that contains a little bit of fly food at one end, a cotton stopper. You put a fly inside that glass tube and then insert it in this channel. On one side of the channel is a light-emitting diode, and on the other is a phototransistor. The LED is producing far-red light, infrared light, that the fly cannot see and won't affect its rhythms. And you have that kind of a design affecting all of these chambers, and we have 30 or 40 of these that we can run on a single computer at the same time back in the 1980s. What this meant was that we could look at single animals in our screen, and we could do tens of thousands of animals in a year or two. So we could, in a very real sense, do a genetic saturation, find all of the genes in this way that could be contributing to this molecular mechanism. And over a series of 5 or 6 years, we found a dozen genes, the most important of which are shown here, that gave us the outline of the workings of this molecular system. And it turns out the system that I'm going to describe for you here is very much like the system that runs our own circadian rhythms, as you'll see a little later in the talk. So the way the system works is that you have these two genes, the original gene found, period, and its partner gene, which we called timeless. And these two genes are activated by two transcription factors, a pair of transcription factors that work together as a heterodimer, clock and cycle, C and B represented here. And when they bind to the promoters of PER and timeless, homologous promoters on these two genes, again on different chromosomes, they activate these genes to produce PER and TIM RNAs. The RNAs are, of course, translated in the cytoplasm to give period proteins and timeless proteins. But then we run into a couple of breaks on this system, delays in this system. The goal is for period and timeless to form a partnership, a per-TIM partnership. That partnership is necessary in order to move these cytoplasmic proteins into the nucleus. If they can get into the nucleus, they physically associate with clock and cycle and inhibit the activity of those two transcription factors. So you have an activation system and you have an inactivation system that is involved in a feedback process. But we have these two breaks. One of the breaks is a break caused by a kinase. This is casein kinase 1, which when we identified the original mutants, before we knew what it was, we called it double time. And double time physically associates with PER. So when PER is first translated, it has two potential partners. One is timeless and one is double time. But double time is produced constitutively, whereas its other partner is being produced with an oscillation. So high levels of double time are always present. So the first partner that period will find is going to be double time. That physical association produces phosphorylations on period, which lead to its degradation. So most of the early forming period proteins are destroyed as they're made by this interaction with double time. At the same time, we have another break in the system which is produced by a photoreceptor called cryptochrome. This is a novel photoreceptor that uses a flavin as a part of the photosystem. When that is activated by light, it binds to timeless, recruits other proteins that ubiquitinate and destroy timeless, and that again is activated by production of light. So during the day, cryptochrome is destroying all of the timeless that's being made. Double time is destroying all the period protein that's being made because there is no timeless, and you won't get this feedback to occur. When the sun goes down, cryptochrome is no longer activated. Timeless can begin to accumulate, and after a delay, period timeless heterodimers will be made. These will move to the nucleus and inhibit, all night long, will inhibit the expression of period and timeless RNAs. So the RNAs will fall and rise in a day-night fashion, and the proteins will fall and rise in a day-night fashion within this system. Now, I don't have time to go into it, but we also know that there are enough delays here that this process will continue to run even in constant darkness, but will be reinforced and synchronized by light-dark cycles, which is why we align, why coming from New York to Naples, we realign our system after a few days. Now let me say a little more about that realignment in a minute, but before doing that, this is a fairly simple molecular process, and of course there's a question of how such a process can inform so many aspects of our behavior and physiology. And in 2001, we did among the first microRNA assessments of the results of having or not having these clocks in Drosophila. And what's shown here are 150 or so gene expression patterns measured over the course of 1, 2, 3, 4, 5, 6 days, a light-dark cycle on the first day, constant darkness for the second day, repeated for a second pair and a third pair to give 6 full days of the record. And what we're doing is each gene expression pattern, we're isolating material every 4 hours for those 6 days, and we're aligning all the results of those assessments for a single gene in a single row. The nearest neighbors in these rows, going from top to bottom through 150 genes, are organized according to the time of their maximal expression, the time of day of the maximal expression. So for example, genes that are down here are at their maximum levels at dusk. Those with maximum levels at noon are in here, and so forth, going around a 24-hour rhythm. So what I hope you see are red and green stripes that represent the oscillating activities of these 150 genes over this period of 6 days. And the oscillations are occurring with roughly a 24-hour interval. But what I want you to notice is that these are not vertical bands of expression. They're diagonal bands. And what this is indicating is that this is not a mechanism that just swings between night and day. There's not just two forms of regulation going on here, night-expressed genes and day-expressed genes. You have every hour of the day unique combinations of genes that are being expressed in response to this circadian system. So instead of being a pendulum, this is much more like a music box, where you play a melody. This whole, I think what we've come to think of this mechanism as one that drives behavior and metabolism through orchestrations of particular gene activities throughout the day and night in a highly specified order. So this system is sitting at the top of gene expression in many, many tissues, as you'll see, determining what the pattern of gene expression overall through a 24-hour interval is going to be. So the last thing I'll say about this is an auxiliary experiment. This is what you just saw. This is what happens when we knock out 3 of the clock genes, timeless period or clock itself. And what you see is the stripes are gone. There's still high and low expression, but it's randomized. So what this is telling us is that, and this is, you'll notice that this is a white and black, these are white and black boxes, which means the data are being collected in a light-dark cycle. So even in a light-dark cycle, which must be the original stimulus, the environmental stimulus that gave rise to this kind of rhythmicity, today, that is not sufficient to produce rhythmicity. You have to have a clock. If you have mutations of the clock, you will not get organized gene expression, temporally organized gene expression. You have to have a clock to interpret the environment. Now, 3 billion years into the development of this system. So now the clock is essential for the flies being able to communicate or to take information from the environment. Now, I thought I'd throw this in a little bit because, again, coming across several time zones to Naples, I'm much like this body-conflicted mouse down here in the bottom of the slide. Once you have molecular probes that you can use to ask where are these clocks, one of the big surprises was they're everywhere. They're not just in the SCN. They have a tremendous role in the SCN in organizing locomotor behavior, but you also find them in muscle cells, skeletal muscle, the lungs, the liver. They're found in essentially every tissue. There are very few tissues that have been found that don't contain working clocks. And these clocks are semi-autonomous in their activities. If you dissect the tissue and you put it in to tissue culture, you'll find that the clocks in the tissue will continue to operate. And of course, you've got this all going on in an intact animal at any time of day or night. So ordinarily, if we have a mouse, for example, that is living in a laboratory in a light-dark cycle, given ad lib access to food, the animal's nocturnal, it's going to be active at night, it's going to sleep during the day. All of its clocks in all of these tissues are going to agree. But of course, the real question is, why do they agree? Do they agree because the environment is consistent, or do they agree because you have a master regulator that is telling all of these clocks what time of day it is? And it turns out to be a little bit of both, but a surprising role, a surprisingly independent level of function for all of these peripheral clocks. So this was discovered by Mike Menaker and Uli Schibler in experiments done about 15 years ago, where they took a mouse that was living on New York time. The light-dark cycle was appropriate for New York time. But instead of having ad lib access to food, these animals had access to food only for an hour in the middle of the day, which this is right in the middle of their episode for sleep. So they would have to wake up if they wanted anything to eat, because that's the only time food was coming into the system. So after a few days, the animals would generate a new routine where they would wake up an hour before the food's delivered, eat the food, go back to sleep, but then get on their running wheels all night long and show a regular pattern of running. When the clocks around the different tissues of these animals started to be assessed, what was realized was this kind of a meal conflict produced changes in the phases of clocks in the liver, in the lung, in the skeletal muscle, but not in the brain. So the SCN continued to follow the light-dark cycle, but all these other clocks started following something that was either the food or a mix of the light-dark cycle and the pattern of food delivery. So the reason I bring this up coming from New York is I think this is very much what happens to us when we travel halfway around the world. When I cross the Atlantic to come over here, this will eventually revert to something like this after consistencies in the environment that last for several days. But for the first few days, I look like this. So I still look a bit like this. Another way of thinking about this is on that trip across the Atlantic, I've left my liver, somewhere in the Caribbean. The Canary Islands has my skeletal muscle, so forth. You're leaving an array of tissues behind along this trip. So I can't stress enough the importance of trying to realign these clocks. Chronic disalignment of this sort, we know from animal studies, can be quite harmful. So let me switch to humans, because I do want to emphasize, it was said before, as you heard, we began this work with Drosophila, but we realized that these genes are also the same genes are present in us. They're present in mice, and they're present in us. And we thought perhaps a good way to understand how these are working in humans is to look at disorders where there are dysfunctional aspects to sleep. So this was just drawn by colleagues at Rockefeller. Could be a student cramming for finals in the middle of the night, but in fact these are individuals with delayed sleep phase disorder, DSPD, typically have a pattern of sleep in which they have very late bedtimes and tremendous difficulties waking up in the morning. And here are some of the features of DSPD. So it's abundant. About 5% of the US is thought to be affected by DSPD. There's a persistent delay in the timing of the major sleep episodes. So you can think of these as night owls. Many of you may be night owls. But strong resistance to efforts to advance sleep phase, which means this is not seen as voluntary. This is not something where you cram for exams and then you catch up on sleep later. This is a lifelong, chronic pattern of sleep and wakefulness. An example of this is shown on the next slide. So you see records from two individuals here. The one at the top is an individual with a normal pattern of sleep and wakefulness, and down here is a DSPD individual that we've been studying. This is double plotted data, so the activity shown on this side is replotted also with a one-day shift on the right side, so that you can see clearly the interval during which the person is sleeping. They're wearing an activity watch, which translates into these bars of activity, much like the running wheel activity that I started off the talk with. But then you see there's this, there are also data coming from a sleep log. The red triangle is in bed time. The blue triangle is, they put a mark in their logbook getting out of bed in the morning. So you see a very clear, regular pattern in the control individual. Sleep onset is about 11:30 in this individual night after night, and wake time is 7 or 8 in the morning. This is a much more chaotic pattern in this individual who was identified next door at Weill Cornell Medical School in this study. And again, if you try to align the reds and the blues to figure out when this individual is getting in and out of bed, it's quite difficult, but you do at least see that this is an individual that gets into bed well after midnight each night, and often is getting out of bed at times approaching noon. So this is a severe sleep-delayed individual classified as DSPD. Now, this individual had a family that was interested in the study we were doing and wanted to participate, and we were able to genotype members of that family by doing whole exome sequencing on the entire family and coordinating those data with data on sleep patterns. So all of those that are indicated in the dark coloring, black coloring, are affected, show an affected phenotype, have a delayed or aberrant sleep pattern. We had one cousin over here, fortunately, that didn't have an affected pattern. And so we gave this individual we assumed that this individual would not be a carrier, and that these individuals could be carriers of a to be identified mutation that was behaving as an autosomal dominant by this pattern. So doing whole exome sequencing, we found a candidate for this effect, and it was a familiar gene from our work with Drosophila and with mice, cryptochrome. This is the Cryptochrome One Gene, and this family carried, the affected members of the family all carried a single nucleotide change in their Cryptochrome One gene in one of the alleles that removed a spliced donor site that was downstream of exon 11 in this 13 exon long transcription unit. Now, what that predicted was that exon 12 would not be able to find a spliced donor site to fuse to exon 11 in making messenger RNA, and in fact would have to jump up to exon 10 to form RNA and protein. And that predicted that there'd be a 72 base pair, 72 base long change in the messenger RNA and a 24 amino acid change in the protein. And here are a number of controls and our one individual that was a carrier, number 11. What you see is that they have two forms of the RNA that differ by 72 bases, and 2 forms of the protein that differ by 24 amino acids. So in fact, these are cells, these are skin, these are dermal fibroblasts from the individual that were showing that this individual was really making 2 different forms of cryptochrome. One of the ways we could, aside from the whole exome sequencing, map the significance of this change is to put that form of the protein coding sequence back into naive fibroblasts that don't have any cryptochrome to drive rhythmicity. So those fibroblasts, in the absence of any additional gene, would be arrhythmic. But we could give them 2 different forms of cryptochrome. We could give them the wild-type form of cryptochrome, or we could give them this new mutant form of cryptochrome and look at the patterns of rhythmicity to see if there were differences that would arise in those cultured fibroblasts. And what you see down here is the result when they're given the full-length, multiple experiments where they're given the full-length version of the gene. And here is the result when given the mutant form of the gene, which produces this truncated protein. So there's about a half an hour difference in the period length measured in these cultured cells in vitro, in tissue culture, indicating that this one gene does have a significant impact on the running of the circadian clock at the level of individual cells. So this increased our confidence quite a bit that this was the mutation involved. So although there's a great deal of homology between the fly clock and the human clock, there is one important difference, and that is instead of period forming a partnership with timeless, in mammals, including us, period and cryptochrome are physical partners to produce that inhibitory complex that shuts down clock and cycles called B-mal in us. So we have much the same pattern of activation and repression, cycling activation and repression, but we now have a per-cry partnership instead of a period timeless partnership. This suggested to us that we should look at the performance of this new form of cry with respect to interaction with per and interaction with clock and B-mal, which we do here. These are immunoprecipitation experiments done in transfected mammalian cells. And what we're doing is we're putting in either the full-length protein, cryptochrome protein, the truncated cryptochrome protein, or a synthetic heterozygous combination of the 2 forms of the protein. So, for example, this is the input. These are 3 different input versions of the protein to do an immunoprecipitation in which we're going to ask what happens if we immunoprecipitate clock in the presence of these proteins. And what you see here is that the full-length protein, full-length cryptochrome protein, is very weakly immunoprecipitated. The truncated form of the protein is robustly recovered. And in the synthetic heterozygote, we almost exclusively produce, we predominantly recover the truncated form of the protein. Now we see very much the same thing if we switch things around and immunoprecipitate B-mal instead of clock. If we instead immunoprecipitate period, the direct partner for cryptochrome, this is the input, and this is what we pull down. So there's no problem. There's no change in the interaction of cryptochrome and period, but there's a robust change in the way that complex interacts with the activators, clock and cycle. So this seems to be dominant, most likely because of this high affinity, a higher affinity of the mutant form of the protein for the targets, the activators in this system. It's as if the truncated form of the protein pushes the wild-type form of the protein out of the picture. So one of the things that we see, you'll notice here that one of the things we've looked at is in transfected fibroblasts, what happens to transcription, to rhythmic transcription in these, in these fibroblasts. The blue plots are plots using the wild-type version of the protein. The red plots are plots of cycles of production in the red Cryptochrome One mutant transfected cells. And what you see here is that for precursors to the per locus, which oscillates because of this system. There's a lower level of expression throughout the 24-hour interval of the clock's activity. Another target, imper1, is also oscillating in a lower range when we have the truncated version of the protein. And a third one, a target that would have been in that microarray, DBP, is also oscillating with a higher amplitude, but in a lower range with regard to that transcription pattern. So it's as if throughout the day there's hyper repression by this altered form of the protein that is attributable most likely to these changes in physical interactions between the inhibitors and the activators. And this is just a control that shows you that for a non-cycling gene, there are no differences of this sort. Finally, just some insights in the way that this process is being seen at the level of chromatin. These are chromatin immunoprecipitation studies in which at times of day of maximal repression, you notice that in red, the cryptochrome transfected cells versus blue, which are the wild type cells, we see a loss of chromatin-associated transcriptional complex. So what we think is going on is the inhibition is being driven by knocking off the activator complex, taking it away from chromatin during times of inhibition. And that's seen on for two different oscillating genes, PER2 and DBP, but not for constitutively producing genes. So this is only having its effect when oscillations are present. Now, one of the big surprises we had early on was the frequency of this mutation in the human population. So as you see, it can be, this is from a database that is carried by the Broad Institute in Boston, in Cambridge. And what you see are the allele frequencies in different populations from around the world. This is 121,000 alleles of whole genome or whole exome sequence that they have in their database, so that's about 60,000 individuals. And the overall frequency is about 0.4% for this allele, but very different frequencies between these populations, with a fairly high frequency, for example, in Europeans, non-Finnish Europeans, of a little more than 0.5%. Since this is dominant, the real frequency is going to be, or the effective frequency for the disorder is going to be twice that of the allele frequency. So about 1 in 75 Europeans will be a carrier, according to these data. So this gave us, this suggested to us that if we could identify fairly large numbers of additional carriers, we should be able to test the correlation of this mutation with altered behavior in a much broader fashion. And we began a collaboration with an investigator at Bilkent University in Turkey who identified, it's now almost a dozen families that had carriers and non-carriers within those families, separating in multi-generation kindreds. So here we're showing a few of the kindreds, just a few of the kindreds, but the results from all of the kindreds shown down here in terms of a clock face with the mid-sleep measurement, that is, when is the individual halfway through the sleep episode, for non-carriers from the same families as carriers. So these are intermixed siblings or relatives from the same, from this collection of kindreds. These are the carriers, these are the non-carriers. What you see is that there's a phase difference between the patterns of sleep between those two groups of individuals, strongly suggesting that this is a highly important risk factor for producing DSPD. So let me summarize what I've said so far. We've got several unrelated families now where this delta 11 mutation predicts DSPD. Should be pretty broadly represented worldwide from the data I just showed you. Since it's found in multiple members of each family, we know it's being inherited. These are not de novo mutations. So we also have to expect that every tissue in these individuals is affected. They have clocks that are running at the wrong clip in all tissues. It's a strengthened transcriptional inhibitor from many of the studies that I've shown you. The competitive binding to clock and BMAL suggests that the dominance of the trait is due to this altered binding of the inhibitors for the transcriptional activators. And I've shown you that expression of just this altered gene in naive fibroblast cells is sufficient to alter the period length of those cells. Now, this is an updated database, and you'll notice that the number down here shows that the total number of exomes now in the database is enough for about 140,000 individuals. So we have 140,000 individuals, and these would be then the frequencies of this allele in all of these different populations. You see some of them can be quite high, which could help enormously in future studies, but it also points to another important conclusion, which is that if you have a database that is that large, then any significant, clinically significant genetic change that would produce altered sleep should be represented in that database. We don't need to wonder what kinds of polymorphisms are out there. We can look in this database. You can run down a list of mutations in these databases, and you can suspect or not suspect different variations that have been seen, understand their prevalence, and hopefully learn something about their impact on behavior. So as I point out here, for most genes, every 3 or 4 nucleotides, there's a polymorphism. So it's a very dense map. And these are single nucleotide changes that have been mapped. You've got allele counts. You've got population-specific carrier frequencies. So what we're doing now is we're focusing on polymorphisms that are both prevalent and predicted to make an impact on the protein, so that if they're large enough, if the frequency is high enough, we should be able to get sufficiently large numbers of subjects to determine the sleep or circadian phenotype. And then, of course, the molecular mechanisms, I think we can explore in cellular models, as we did for this last DSPD mutation. So here's a list of some of the mutations that we're currently investigating, where we have 2 or more families in Turkey that are carrying these changes, which can be subjected to studies in both cell culture and in human subjects. And here's just an example of one of the kinds of assays we can do. We can put the mutant form of the protein coding gene back into a naive fibroblast. Here we see no change in the rhythmicity, indicating that the polymorphism is probably not going to give us a change in the subject pattern of sleep or circadian rhythmicity. Here, on the other hand, is a mutation which is producing arrhythmicity. So that's going to be carried as a heterozygote in the population. We'd have to look at those subjects to understand whether there's a dosage, whether that change in dosage can produces a phenotype. But those are the kinds of steps we would take. Now, another point, though, is to look at this a different way. Again, all the cells in the body are carrying these, so are there other important aspects of metabolism and behavior that will be clinically important and studied in this fashion? So if you have a high allele frequency, if you can identify large numbers of subjects in multiple families, then you can, if you see comorbidities for a sleep disorder and a metabolic disorder or a psychiatric disorder, for example, you should be able to with confidence link that specific genetic change to that other disorder because of the high degree of reliability of the assessments coming out of multiple families. So we think this kind of a backwards approach of using the databases to find phenotypes and then have confidence that the phenotype is due to that particular mutation is quite high. And I'll just give another final example, which is we know from mice that Cry1 is a central regulator of hepatic gluconeogenesis. If you overproduce Cryptochrome 1, the gene that we've been talking about, this blocks glucagon signaling, retards glucagon signaling in the liver, producing hypoglycemic mice. And you've got oscillating Cry1 in those mice periodically affecting gluconeogenic gene expression in synchrony with feeding and fasting cycles. So if we've found a dominant gain of function in that same gene, we have this question of whether or not subjects that are carrying that mutation also have problems with regulation of their glucose. So does the Cry delta 11 mutation alter glucagon signaling in these human carriers as constitutive overexpression would in mice? So just to point out who did all the work, the actual investigators as opposed to the principal investigators. Scott Campbell and Patricia Murphy were collaborators at Cornell that identified our first group of subjects for studying DSPD. Anna Krieger helped us with the analysis of their behaviors. Tayfun Osalek and his student Onur Anat at Bilkent University identified families containing this mutation for us. And this is Alina Potke, who did all of the human molecular biology that I've been describing in our lab at Rockefeller, and of course here are our sources of funding. But finally, as you heard earlier, I think we don't want to forget that all of this started with the fly. So you can get to humans in good time if you have a strategy. We'd never be able to find this system starting with humans. We had to start with a simpler organism. But starting with a simpler organism doesn't tie your hands. There's this opportunity, especially in contemporary science, to move easily between these systems. You can move easily back and forth between something like Drosophila and human studies. And I think this brings us a much deeper appreciation of these systems in the long run. Thank you very much.